Hello, and welcome to Virtual Investor Conferences. On behalf of the Life Science Investor Forum, we're very pleased you joined us for our next presentation from Percheron Therapeutics. Please note you can submit questions for the presenter in the box to the left of the slides, and you can view a company's availability for one-on-one meetings through the Book a Meeting tab found in the top toolbar. At this point, I'm very pleased to welcome Dr. James Gardner. He's the Managing Director and Chief Executive Officer of Percheron Therapeutics, which trades on the OTC markets under the symbol PERCH and on the ASX as PER. Welcome, James.
Thank you, John, and thank you for the opportunity to be here today at the conference. It's wonderful to have the opportunity to introduce our company. And I'm going to jump straight in and talk a little bit about what Percheron does and look forward to addressing some questions at the end of the presentation. Just to begin with the high-level points here, we're a biotech company and we're focused primarily on oncology. We have a lead drug candidate in development called HMBD-002. And that's what I'm going to be talking about today. And this is a drug that has potential applicability to a range of different cancer types. It's one of these drugs that we might be able to use in a number of different tumors. And the great news is that it's already completed phase 1 human trials. We've done all that initial work to understand the safety profile of the drug. And what our company is now focused on is taking this drug into what we call phase 2 human trials, where we really get to find out whether the drug works and how it works. So this is an exciting time for our company. It's an exciting time to share it with investors. And we think there's some really interesting things ahead for us. I'll jump ahead a little bit here and double back on some of this material, but I just wanted to give you a very quick sense of the team. We're a very lean company, but our board is comprised of some very experienced drug developers, veterans of 20, 30 years experience in drug development. And so although our company is really a virtual startup in shape, we bring a lot of that track record to the business. And although we're a small company, we're reasonably well financed. We closed our most recent reporting period as at 30 June 2025 with $10.6 million at bank. And we run very, very lean. The vast majority of our cash is spent purely on the R&D work. We have very, very low overheads. We are an Australian domiciled company, and for those that have spent a lot of time in the biotech sector, one nuance of this is that we do benefit from a tax rebate through the Australian government. They will pay us back up to 43.5 cents on the dollar for every dollar we spend on R&D. And that's been a very, very useful source of funding for our company. It fundamentally means that every dollar that investors put in is matched up to 43.5 cents in the dollar by the Australian government, and it's a wonderful extra source of funding for companies such as ours. I'm going to jump into the science here and talk a little bit about what we do. And the first thing to say as I start to talk about our drug, HMBD-002, is that we sit in a very, very interesting space in the treatment of cancer, which is where the immune system plays into cancer. One of the most important realizations over the last 25 years or so has been the very, very important role that the immune system plays, both in the genesis of cancer and in our response to it. Ordinarily, all of us in our course of our daily lives are generating pre-malignant cells, and normally our immune system removes those in just the same way that it removes bacteria and viruses and so forth. For a cancer to become established, for a cancer to take root, so to speak, it needs to evade the immune system. It needs to suppress the immune system. And one of the things we've discovered over the last 25 years is there's a number of ways that cancers do that. And they do it primarily by virtue of protein molecules on the surface of the cancer cell, which interact with matching proteins on immune cells. And the interaction between these proteins has the effect of producing an off signal for the immune system. They damp down the immune activity. Now, there's a couple of these signaling interactions which have already led to FDA-approved drugs. And you can see some of those drugs shown on the right-hand panel of the slide here. The 800-pound gorilla in this field is a drug called Keytruda, also known as pembrolizumab, manufactured by Merck. This is a $30 billion plus per annum product. But there are quite a number of these other protein interactions. So one of the things we've been looking at over the last decade or so is how we can potentially drug some of these other interactions, stop the cancer from switching off the immune system and reactivate it so that it can fight the cancer cell. Now, one of these interactions is a molecule shown at the bottom left of this slide, a molecule called VISTA. And VISTA is the target of our drug. It is the target of 002. Our drug is, in effect, a VISTA inhibitor. So what we're trying to do is to inhibit VISTA, reactivate the immune system so it can go and fight the cancer on our behalf. Now, why VISTA? Why are we targeting that particular protein? Well, there's a number of really good reasons why this looks like a very, very promising target for cancer therapy. As you can see on the left of the slide, VISTA is widely expressed on a huge range of tumors. It's a very, very ubiquitous target. It is more widely expressed, for example, than PD-1, which is the target of Keytruda, the drug I mentioned a moment ago. And so by targeting VISTA, it gives us the potential to target a very, very broad number of cancers. In general, as is reflected in the middle panel of this slide, the more VISTA you have in your cancer, the worse your prognosis. And so it stands to reason that being able to target this would help us. And then on the right-hand panel of the slide, you see something that really intrigues us, and that is the fact that in patients who are resistant to drugs like Keytruda, it appears that VISTA is part of the resistance mechanism. It appears that this protein is relevant to the way in which tumors become resistant to drugs like Keytruda and others that are similar. And so it gives us a really, really tantalizing possibility to try and extend and enhance the use of drugs like Keytruda. And we think that may be a very, very important opportunity for our drug. I'll jump ahead a little bit here just to give you a sense of where the drug is in its development. I think any veteran biotech investor will be familiar with the fact that this is a long haul to take a drug from the bench to the bedside, and it's failure prone. A lot of things can go wrong along the way. And I think one of the things that really appeals to us about this drug is that it has been through quite a large part of this journey already and has cleared many of these hurdles. In the top left of this slide, you can see it diagrammatically illustrated some of the important lab work and preclinical work that has to go on in many cases before a drug can even be first used in a patient. We have to, for example, test its toxicity in animals. We have to do a lot of that kind of work. And the great news here is almost all of that has been done. We may still have a little bit of work to do along the way, but we think we can do that in parallel with the clinical trials. And so far, it appears that the drug is extremely safe. We get very, very little toxicity when we use it in animals. And of course, for a cancer drug where we're using it sometimes in very sick patients, sometimes in combination with other medicines, that safety profile is all important. If you look at the bottom of the slide, the purple section here, we've already learned how to manufacture this drug. It's a particular kind of drug called a monoclonal antibody. And monoclonal antibodies are not trivial to manufacture. This is still quite difficult work to make these drugs well and to make them consistently. And it's something that people often forget about in biotech. But the reality is when you come to FDA with a new drug, FDA really just wants to know three things. They want to know, does it work? Is it safe? And can you make it? And I think for this drug, we have largely answered that question. We've made multiple batches very consistently, and we get a pretty clean product out the end of it. So we're pretty comfortable now with the manufacturing. So what that leaves is just the top right-hand part of the slide here, which is the clinical trial work, the human trials to show, first of all, is the drug safe? And secondly, does it work? And we've completed phase 1, which is really the safety part of that equation. And we're still receiving data from this study. We haven't yet announced all the results. That'll be coming in the next month or two. But on the high level evidence so far, it does look like the drug is very, very well tolerated. We see very little toxicity with 002. And as I say, for a drug in cancer, that's very important. Now, when we just zoom in a little bit on some of that preclinical work that I touched on, we've tested this drug in quite a wide range of animal models of cancer. That the health warning that always comes with these animal models is they're never perfectly predictive of what happens in the more complex environment of a human patient, but they give us a really good sense of whether our drug is moving the needle or not. And consistently, in almost every animal model that we try this drug in, it seems to have a meaningful benefit in terms of slowing tumor growth. And as you can see on the right-hand side of the slide here, we've done a few experiments where we've put the drug into animal models of cancer in combination with pembrolizumab, with Keytruda. And when we do that, we see an incredible synergy between the drugs. It almost completely shuts down growth of the tumor. So we think this is exciting data, and we think this gives us a really good basis to take this drug into clinical trials. And I should say there's a wealth of data behind all this. I'm just showing some high points here, but we've done really a lot of work to understand the potential uses of this drug in humans. I mentioned that we completed a phase 1 human trial. Again, just to manage expectations, phase 1 studies are safety studies. They're really designed to tell us whether we can dose our drug and how high we can dose it. This study was done at 6 centers in the United States, as you can see listed on the right-hand side of the slide, under, of course, the oversight of the FDA through an IND. And we went up through 7 doses of the drug by itself as monotherapy. We actually did not establish a maximum tolerated dose, which is the usual outcome of a phase 1 study, simply because we just didn't see enough toxicity. We continued dosing the drug well into what we anticipate to be a therapeutic range, and we just didn't see a lot of dose-limiting toxicities, as we call them. So actually a very, very strong safety profile that emerges from this work. In this phase 1 study, we also looked at 4 doses in combination with pembrolizumab. As you can probably start to tell, this is something we're really interested in exploring. And again, the drug seems equally safe in combination with pembro. And in fact, we've shown that diagrammatically here. You can see in this little bar chart on the left of the slide, you can see the toxicities associated with our drug that as a monotherapy, and then in the second bar, toxicities in combination with pembrolizumab. The 2 bars look almost identical. And what that reflects is there's really no kind of additive toxicity. And I always think it's important to just qualify this kind of data in a phase 1 setting. All the patients in this study had our drug, and clinicians are always looking out for every possible toxicity, but some of the things they pick up are potentially effects of the cancer rather than the drug. Things like fatigue, for example, are very, very common effects of cancer. And so as we move forward with the drug, as we learn more about it, we'll probably find that even some of these more mild toxicities actually start to fade into the background a little bit. This phase 1 study was done in advanced cancer patients. And when I say advanced cancer patients, these were very, very late stage patients. Many of them had had 3 or 4 or more lines of previous treatment. They'd had a lot of different cancer medicines thrown at them. And it was a very, very mixed group of patients, as you can see from the bar chart on the left, which again breaks down monotherapy and combination. We had some breast cancer patients, some bowel cancer patients, some pancreatic cancer patients. But as is usual practice in these phase 1 studies, it was a little bit of everything in the study. And that makes it really hard to pick out any sort of efficacy signal and to say, well, here's where the drug appears to work. But we did see certainly some individual patients, some individual case studies, if you like, that gave us, I think, a really good signal that at least for some of these patients, the drug is moving the needle. And you can see some of these patients itemized here. To give you some context, 18 cycles of treatment, which is what we saw with the patient in the top row there, that's a little over 1 year on drug. And to put it very candidly, patients at this stage of the disease, at this very, very advanced stage, typically don't remain stable for a year unless they're receiving some sort of benefit from a medicine. So this sort of data certainly gives us some pointers that our drug is beneficial. To be clear, we can't hang our hat on this. This is a signal rather than a result. And what we need to do now is to take this drug into a phase 2 study where we can really try and drill down on this and quantify the benefit that it provides to patients. So what does that phase 2 study look like? Well, we're planning to launch the phase 2 study next year, calendar 2026. And right now we're in a process of talking with clinicians and researchers and advisors about exactly how it should look. And in due course, we'll take those conversations in all likelihood to FDA. But there's a few different angles and a few different themes we might look at here. One is to look at the drug by itself as a monotherapy, as we call it. And there's certainly some potential use cases for the drug there, but we're actually really interested in potential combination use here with existing immunotherapy drugs like pembrolizumab, but also potentially in combination with other kinds of treatment. We saw some preclinical data published very recently from a team at Stanford University in Palo Alto, California, where they combined our drug with radiotherapy and actually saw some very, very promising results there. So some of those kind of angles we're really investigating as well, because we think our drug has potential to overlay on quite a broad range of different cancer treatment modalities. Now, one of the things that people often ask for any drug in development is how well it fits into the competitive landscape. How does our drug compare? Because almost always there are other people working on the same target, working in the same sort of space. And there are other people working in our space too. There are other people producing drugs against VISTA. And we've shown on this slide some of the key ones that we've been tracking. Now, the reality is that many of these programs have been discontinued during phase 1. And certainly what we hear anecdotally is that a number of them have been discontinued quite specifically because of a phenomenon called cytokine release syndrome. Cytokine release syndrome, very simply, is a kind of overactivity of the immune system. And it's an occupational hazard when we're giving drugs that are designed to stimulate the immune system, because obviously we can stimulate it too much. We can turn it on and cause all sorts of other issues for a patient. And so we understand from talking to clinicians that a number of drugs targeting VISTA have been limited very significantly by cytokine release syndrome. We have not seen a single case of cytokine release syndrome with our drug. And we've had it now in 48 patients at various doses. We've got a pretty substantial body of clinical experience, 0 cases of cytokine release syndrome. And that's not by accident. That's not good luck. It's because actually our drug, 002, is a very slightly different kind of antibody from just about anything that's been tested in humans. As you can see from the slide, most of the other drugs that have been tested here are of a type called IgG1 antibodies. And I won't go into a lot of detail about what that means, but they're a more aggressive kind of antibody. They're more potent activation of the immune system. Our drug does not have quite the same degree of activation as an IgG4 antibody. It blocks the VISTA signaling just as well. It blocks those protein-protein interactions every bit as well as any other antibody, but it doesn't draw in immune cells to attack the cancer cell. And that's why we think that we're not seeing cytokine release syndrome. And it does mean that we have something fundamentally different from just about every other drug in this VISTA space. And we think that's a really important point of differentiation for 002. Where do we go from here? Well, I'll be very transparent and say that our next step is to do the phase 2 study, which we hope to start next year. We are unlikely to take this drug all the way to commercialization ourselves. We're a pretty small company, and we think that sometimes that job is done better by larger companies. So somewhere along the way, we will be looking to partner this. Now, what could a drug like this be worth in a partnering transaction? Well, clearly individual mileage varies. Every partnering transaction is unique. But I've shown on this slide some recent benchmarks for deals with loosely similar programs. The early to mid-clinical deals are sort of reflect where we are now, and the late clinical deals, where we could potentially be in a couple of years' time off the back of positive phase 2 data. So we think there is a very, very real and substantial commercial opportunity associated with this drug. But as I say, our ambition as a first approximation is to put it in the hands of a larger company for commercialization. So I'm going to stop there, and there's a couple of questions that have sort of come in while I've been speaking. So I'm going to just spend a few minutes now addressing those, and please feel free to enter some more as we go. The first one that's come in is in relation to funding requirements and how we propose to fund the phase 2 study. And I can only obviously speak very generally to this point. It's obviously one that's front of mind for investors almost always. But as I say, we closed our most recent quarter with a little over 10 million Australian dollars at bank. And we have since received, through the Australian government R&D tax rebate mechanism that I mentioned earlier, about 1.6 million Australian dollars, about 1 million US in the last month or two. So we continue to benefit from that scheme, and we anticipate being able to continue to benefit from it in the future. So that very, very significantly reduces our cash requirements. We're looking at some novel study designs that will hopefully allow us to do a lot with not a huge amount of investment, and very difficult to say very much more than that at the moment, because the study design is still work in progress. But we do think there's a lot we can do potentially with the resources we have. So we feel that we're not short of money at the moment. We feel that we're not restricted in being able to move the drug forward. But obviously, like every pre-revenue biotech company, that's an ongoing matter. There's a question around why the drug's original developer has licensed it to us. And just to provide some context to this, I should say that HMBD-002 was not invented by our company. It was invented by a privately held venture-backed company who licensed it to us earlier this year. And these kinds of licensing transactions are common in biotech. Many drugs go through 3 or 4 owners by the time they reach market. And but obviously it begs the question in investors' minds why the originator would let go of such a promising drug candidate and pass the baton to a company like ours. And I think in this case, the reason is simply that the company that originated this drug, a company called Hummingbird Bioscience, is really a company focused more on kind of discovery and early stage development. They have an incredible discovery research lab, incredible capabilities there. They are, in my view, a potential world leader in areas like antibody drug conjugates. But I think the mid to late stage development is perhaps not in general where they focus their business at present. So very often in these situations, companies that are more discovery focused pass drugs to companies that are more development focused during their genesis, and I think this is a case of that. And then finally, one last question here is around upcoming milestones, and we sort of glossed over this a little bit in the presentation, but I'm going to double back here and just put up a slide we have in our presentation. By the way, the presentation here will be available through the OTC Markets platform, and it is available through our website. Everything I've presented here is publicly disclosed. You can download it from our website. But there's some things coming up which I think are relevant for investors and which I think would be good for people to keep an eye on. The first is that we will be getting full and final data from our phase 1 study over the next, let's say, 6 to 8 weeks. And as I said at various points through the presentation, phase 1 studies are safety studies. This is not going to, in all likelihood, give us a blinding revelation of multiple patients being cured, but it is going to give us some really good granular understanding of the safety profile of the drug. It's going to give us a greater degree of confidence in its activity, and it's going to give us really important understanding that goes through into phase 2. So I think that's an important milestone for the drug. We do plan in the fourth quarter of this year, fourth quarter of calendar 2025, to come out and share much more details specifically about what we plan to do in phase 2 next year. As I mentioned, that's an ongoing discussion right now. We're talking to clinicians and researchers and experts about exactly how we should approach this. But we're drawing those thoughts together, and we plan to share those with investors over the next couple of months. And I think that'll be an important milestone for the company, because I think at that point it becomes very, very clear what the path forward for this drug is. And then, all being well, our aspiration is to start that study in calendar 2026. And I think that's really where the rubber hits the road for biotech companies, is once we get back on the saddle and back in the business of running clinical trials. So that's something that we're aiming to do next year, and I think that's going to be a very, very exciting year ahead for our company. So, ladies and gentlemen, with that, I'll just say thank you very much for your attention and for the opportunity to present here. As I say, there's a wealth of additional information on our website, and please do feel free to book meetings through the conference platform. We'd be delighted to discuss this and other points over the course of the conference. Thank you very much indeed.